Hello everyone! In this lecture we're going to talk about some material from chapter 2 of our text. In this chapter we focus on the structure and the function of neurons or cells within our nervous system. Now let's take a look first at the division of our nervous system, and here we are looking first at the central nervous system, or the CNS for short. That includes our brain as well as our spinal cord, and we can see that here on the right hand side of the slide. Both the brain and the spinal cord are encased in bone for protection. So your spinal cord is encased within your spinal column, and your brain, of course, is encased in your skull. Then we have the PNS, or peripheral nervous system. So this is going to be stuff that's outside of the bone. So this will be nerves and sensory organs like your skin. Now let's look at the basic units that compose the nervous system. Here we're going to focus on neurons, or nerve cells, and we're going to talk about briefly the different types of nerve cells, and we will revisit this later on in the course. But also we're going to mainly focus in this lecture on the structure of a basic neuron. And again, neurons are nerve cells. So these are cells within your nervous system. You have billions of these. The goal of a neuron, or the purpose of it, is to process and transmit information. So for example, when you sense something from the environment, that signal from the environment from your sense organ, for example, your skin, has to be sent to your brain, right? So nerve cells are what aids in that process. They're what makes that possible. Of course, as you might imagine, there's lots of different types of jobs that nerve cells would have to do. So what we have are specialized neurons. So we have different neurons for different jobs, and their structure is in turn different. Now, some things most neurons all have in common are the cell body, or a soma, and we can see that here on the right-hand side of the slide. That would be the cell body, or the soma. And also, just to take note that the cell body is going to vary in shape depending upon what type of neuron it is. But again, most neurons are going to have a cell body. Also, within this cell body, or soma, the nucleus will be contained within that. And the nucleus, just to kind of remind you, I'm sure most of you all are at least familiar with the nucleus, but the nucleus within that is contained a lot of what we call machinery that provides life processes for the cell. That is energy and things that the cell needs to survive and not die off. Those things are produced within the nucleus. So, as we said in the last slide, as we discussed, most neurons have a cell body. So let's take a look more closely at the cell body, or the soma. So first, let's take a look at what's around or surrounding it. So we'll take first a look at the cell membrane around the nucleus. So here we have our soma, or cell body, and within that here we have the nucleus. So here within, where we see this purple outline here, this is the cell membrane, and again this is going to be protective. It is composed of what we call lipids, so a double layer of lipids. These are fat cells. And there are specialized proteins within the cell membrane. So protein molecules embedded within this double layer of lipids, or fats. And the point of this is to detect outside substances. So things that are out here outside of the cell body. The goal of this membrane is to first encase everything within the cell body, but then also to provide a protective layer from what's on the outside. So if there is a substance out here, this membrane can detect it and keep it from entering the cell. And as I just stated, of course, controls access to the cell's interior. So again, it's going to keep unwanted items from the outside from getting into the cell's interior. In turn as well, though, while it can prevent things from getting into the cell that shouldn't get into the cell, it in turn can also allow things to leave the cell. So it can transport certain molecules in and out of the cell that do need to leave and then come back into the cell. So it kind of serves both functions, to keep things out, but also to allow things that need to come in to come in. Now, focusing more on what gives a neuron its structure, we have to take a look at the cytoskeleton. The cytoskeleton is really kind of the actual skeleton of the neuron. It's what gives it its shape. It's much like our skeleton, for example. The cytoskeleton is composed of a lot of protein strands, kind of like a matrix of protein strands inside the cell body, and we can see that here. And again, this is made up of a lot of different protein strands. And then, and again, the function is to give the neuron shape and to help the neuron maintain its shape. There are 3 types of protein strands that compose the cytoskeleton, and the thickest of which are what is referred to as microtubules. And as you might imagine, that basically just translates to tiny tubes. We can see those here towards this tail end of the cell body. And leaving and moving away from the cell body. If we look within here, we can see these tiny little tubes or microtubules. And what these are bundles, tight bundles of that protein. So protein filaments. There's actually 13 protein filaments here. And these are kind of built around a hollow core. And the core here is hollow because it's going to allow for substance transport. So, for example, things leaving the cell. And we'll get more into that as we move along as well. So now that we've discussed what's going to give the neuron its shape, and we've also talked about that outermost layer, that protective layer of the membrane, we're going to now look inside the membrane. We're going to look at the different structures within the cell body. So first, let's take a look at the ER for short, or endoplasmic reticulum. This is going to be surrounding the nucleus within the cell body. So if we look closely here on the right, we have our nucleus, and then surrounding the nucleus, we have the endoplasmic reticulum. We have both rough here, and then outer more, we have smooth. Now what the ER does is first encase the nucleus. Both of these contain membrane that encloses the cell or encloses the nucleus. The rough ER contains what are called ribosomes. And what these do is these are what produces the protein for the cell or in the cell membrane. So this is where protein production is happening. And the smooth ER, this outer portion of ER right here, this is going to provide channels for segregation. So keeping certain molecules separate for different processes that the cell needs to go through in order to survive. Also, lipid molecules or fat molecules are produced here. Next, we're going to take a look at the Golgi apparatus. That is located right here next to the rough ER. And the Golgi apparatus is actually a type of smooth endoplasmic reticulum. Now, what happens here is the formation of complex molecules. So within the Golgi apparatus, complex molecules are made from more simple molecules. Also within the Golgi apparatus, products are wrapped. So we call this the wrapping center. Certain things that are being produced within a cell need to be wrapped before they can be transported. So for example, a hormone. The hormone would be encased in the Golgi apparatus and then released out so it can go wherever it needs to go. But again, the product of the hormone itself is packaged and wrapped here. Also, within the Golgi apparatus, there is production of what is called lysosomes. And lysosomes are little sacs containing enzymes for substance breakdown. Sometimes there's extra stuff in the cell that needs to be broken down and gotten rid of. Lysosomes contain enzymes that will make that possible. And those are produced again in the Golgi apparatus. Next, we'll take a look at these bean or bead-like structures called the mitochondria. So these look like, again, oval little beads here. They are made of a double layer membrane. And on the inside, there are little wrinkles that look like shelves. These are called cristae. And within the cristae, we have the breakdown and extraction of nutrients. So basically what happens here is that the cell itself will provide the mitochondria with nutrients. And inside the mitochondria, the breakdown and extraction of basically stuff to provide the cell with energy occurs. The energy is in the form of adenosine triphosphate. Next, we'll look at the jelly-like substance that kind of fills out the inner membrane. So all of these structures like the mitochondria, the nucleus, and the Golgi apparatus, they need to be suspended in something so they don't move around on the inside of the membrane. And they are suspended in the cytoplasm, which again is just this kind of gooey jelly-like substance. It again contains the small structures that we've kind of been talking about, the mitochondria, Golgi apparatus. These are also referred to as organelles. The nucleus here contains the nucleolus and chromosomes. So within the nucleolus, this is where the production of ribosomes happens. So ribosomes are responsible for protein synthesis or protein breakdown. And the chromosomes are what holds the cell's DNA. So the DNA information for the cell is held within the chromosomes inside the nucleus of the cell. And within the chromosomes, there is also production of what is called mRNA, which is basically a copy of the DNA of the cell. So basically a copy is made, and again that is called mRNA, that attaches itself to the ribosomes, and that can then be transferred outside the cell. Now, thus far we've been talking about the cell body or the soma and what is inside the cell body. And again, most type of neurons have a cell body. They also have what are called dendrites, and those are these branch-like structures that are coming off of the cell body here. The dendrites receive messages from neighboring neurons, so signals sent from neighboring neurons all around will be received by this neuron from its dendrites here. Now, I want to emphasize here that this is one-direction communication, meaning that the dendrites can receive messages or signals from other neighboring neurons, but the communication or message that's sent by this neuron that we're looking at right now will leave it through the tail axon here, which we will talk about here in a second. So messages from this neuron that we're looking at here don't leave from this neuron through its dendrites. So the dendrites only receive information, they don't send information. Now, also, most types of neurons have an axon, which I just mentioned in the last slide, and that is that tail-looking structure here that is leaving the other end of the cell body. This is again, like you can see, a tube-like structure. This is often what we call myelinated, and that's what we see here, these bead-like structures. This is kind of a form of insulation for the axon, and it helps the electrical signals that are coming from the cell body and traveling down the axon travel more smoothly and not leave the axon before it gets to the terminal buttons at the very end here. And again, this myelin here, again, that's going to help with the electrical signal or information that's leaving this cell via the axon. And that information or signal, if it is being sent to the terminal buttons to leave the cell to go to other neighboring cells, it travels along the outside of the axon. And lastly, most types of neurons have terminal buttons. These are located at the very end of the axon here. And they kind of look like little knob-like looking structures. These right here, once we have a electrical signal leaving the cell, traveling down the axon and reaching the terminal buttons, once it reaches the terminal buttons, it changes from an electrical signal to a chemical signal. So some neurotransmitter, which we will be talking about neurotransmitters as we move forward in our material throughout the semester, some neurotransmitter, and that will depend on what type of cell we're working with here as to what type of neurotransmitter it will be. But for now, just know that that is basically a chemical that's going to be released from these terminal buttons out into the space or the synapse between this cell and all of the neighboring cells. And depending upon what neurotransmitter is released, the reaction from other cells will be either excitatory or will increase their activity, or inhibitory, which will reduce or inhibit their activity. So now continuing for a second with the terminal buttons, they do need things that are only produced in the cell body. So they will need substances that the cell itself, the cell body, produces. So how do these substances get from the cell body all the way down to the terminal buttons? This happens through a process called axoplasmic transport. Now, that's a big fancy way of saying substance transport. So some substance will be transported one of two directions, either from cell body to the terminal buttons, or the opposite direction from terminal buttons to cell body. Now, the thing to notice here is that we're not talking about substances or signals being sent outside of the cell that we're looking at right now. That is a bit different. That is called action potential, which we will discuss next week in depth. For now, we're focused on this slide, we're focused on substance transport within the cell itself. So what happens when some substance is moving from either cell body to terminal buttons or from terminal buttons to cell body? The microtubule tracts on the inside of the axon is where the substance will travel. So in the previous slide, I mentioned a signal being sent along the axon and leaving the terminal buttons and then going to the space or the synapse here to the surrounding cells. So when that occurs, that is again called an action potential, which is different. But when that happens, the signal is traveling on the surface or on the outside of the axon. In axoplasmic transport, the substance or signal is traveling along the inside of the axon. Now this can happen in one of two directions, like I mentioned. Anterograde is when we have substances traveling from the cell body to the terminal buttons. So when that happens, a protein called kinesin or kinesin attaches to the item or the substance that's being transported and then helps it propel down the axon track here. So kinesin or kinesin would attach itself and then help propel along the inside of the axon all the way down to the terminal buttons. And then, as I've also mentioned, there is the possibility that substances can travel the opposite direction from terminal buttons to cell body, and that is retrograde transport. So another protein called dynein or dynein will carry substances from the terminal buttons here back up the opposite direction of the axon to the cell body, and again traveling on the inside of the axon. And this will happen when the terminal buttons have a substance that they need to get rid of, basically. So it will be taken back to the cell body for disposal. And just a quick note, retrograde transport, it happens about half as fast as anterograde. So cell body to terminal buttons happens much quicker than in the opposite direction. Now we'll look at the type of cells by extension. So these are different types of neurons, and the first one we'll take a look at here is unipolar, uni meaning one. So with unipolar neurons, there is a single process leaving the cell body. We see that here. So here's the soma, cell body, and a single process leaving it. We only see these in invertebrates. They're very common in insects. You won't find this classification of neuron in a human. Next, we'll take a look at a bipolar neuron. So here we have one axite and one dendrite leaving the cell body here. So we have two processes, an axon and a dendrite. And that is typically a specialized type of sensory neuron. The most common type of neuron is a multipolar neuron. It's probably the type that you are most familiar with, and it is also the type that we've been looking at throughout this lecture thus far. And we'll see here it's called multipolar because we have a single axon or single process leaving this side of the cell body, but then we have several dendrites leaving the soma as well. And lastly, we have a pseudo unipolar neuron. So this is basically where we have an axon that is split and has two branches. So we have our cell body, and then we have the axon here leaving, but then the axon splits. These type of neurons are very interesting in that one branch of the axon is going to the central nervous system, whereas the other is going to the peripheral nervous system. So one branch is going to either the brain or spinal cord, CNS, and the other branch is going to the peripheral nervous system or some sense organ. These are commonly found in the cranial nerves, which makes a lot of sense is in that the cranial nerves are what are bringing sensory information into our system. We'll talk about the different cranial nerves later in the semester, but for example, we have a cranial nerve for smell. So basically, there would be one branch of an axon that would be going to the peripheral nervous system that would be bringing in the scent, and then there would be another branch of the axon that would be going to the brain, for example. So this is kind of a unique type of neuron, and as you can imagine, pretty important because it's going to aid in us making some sense of sensory information that we have in our environment. Now we'll talk about some very important cells. We've been talking about neurons thus far, but neurons wouldn't be able to survive and thrive and function without what we call supporting cells. These are cells that basically provide neurons with a lot of different services. So for example, in the central nervous system, neurons are about half of the cells there. The rest of them are supporting cells to take care of the neurons, basically. And these are really needed because neurons need a lot of things. Neurons are very active, so they burn a lot of nutrients. Supporting cells will help with that. So we have different supporting cells depending upon if we're looking at the central nervous system or the peripheral nervous system. So we'll first start with the central nervous system, and we'll look at what are called glial cells or neuroglia. These are also commonly kind of referred to as nerve glue. So basically what they do is hold neurons in place and kind of surround them and buffer them chemically and physically from what's going on the outside. So kind of holding them in place and protecting. They also control a nutrient supply and some chemicals as well. And they also insulate the neurons, so the messages that are being transported from neuron to neuron and within each neuron kind of stay contained on the inside and don't go every which way. They also help to remove dead neurons as well, which cleaning out debris is also incredibly important, and the supporting cells do that as well. Now we have different types of glial cells. The first one we'll talk about are called star cells or astrocytes. So these are really the type that are going to provide that support for neurons. So if you look, you can kind of see why they would be called star cells. They kind of make a star-like shape or star-like form, and you can see here's our astrocyte, and here are a bunch of surrounding neurons on the outside, and this star cell is kind of holding them all some distance apart and kind of holding them in place. Astrocytes also help to clean debris out of the brain. This is commonly called phagocytosis. So again, this is just removal of any debris, extraneous substances and such that don't need to be around. These type of cells are the type that will help clean those out. And they also do help with the chemical composition of the fluid surrounding the neurons, maintaining a balance so that it is a healthy fluid for them to be in. And they also do provide the neurons with some nutrition as well. For example, these star cells can receive glucose and convert it into lactate, and then release that lactate to the extracellular fluid around the neurons. And the neurons can then uptake this lactate and transport it to the powerhouse within the neuron, the mitochondria, for energy. The next type of glial cell in the central nervous system, this is a very important type of glial cell. These are called oligodendrocytes. So what these guys do is really support the axons of the neuron. Specifically, they create the myelin that wraps the axons. So here we're looking at an oligodendrocyte, and here we have a few neighboring axons, and what you'll see are all of these structures coming off of the oligodendrocyte, and they're wrapping around the axons and forming these beads of myelin. So these are like little paddle-like looking structures, and they, again, the paddles themselves become the myelin of the axon, and the different beads of myelin are about 80% lipid or 80% fat and 20% protein. And again, you see that these are like segmented or bead-like looking structures. The spaces in between them are called the nodes of Ranvier. Interestingly, a single oligodendrocyte can produce 50 segments of myelin. And again, this is going to happen when the little paddle-like process leaving the oligodendrocyte wraps around the axon. And again, this is in the central nervous system, and this is really happening when the central nervous system is developing. The last type of glial cell is microglia, micro meaning small. And you can see here, this is an example of a microglia. Overall, it is small, and then it is irritated in some way, and it will kind of puff up and become inflamed. And eventually, it will wind up looking like this, a completely different structure. This is very, very important because what this inflammation really does is kind of a protective response. So for example, if you have some sort of brain damage, the brain inflammation or the brain swelling that we see, the microglia are what is responsible for that. And again, that is a protective response that the brain does, is to inflame itself when there has been damage. These glial cells, these type of glial cells, microglia, also do help with phagocytosis, or the clearing out of the debris from the brain. They also do perform a protective function to try to protect the brain from invading substances that don't need to be hanging out. So we've talked about the supporting cells in the CNS. Now let's talk about the ones in the peripheral nervous system. So we have Schwann cells. These are basically just like the oligodendrocytes in the CNS, except they are in the peripheral nervous system, and they perform their function a bit differently. So they also help myelinate the axons in the PNS. But the way that they do it is they completely surround the axon. So the pink portion here is an axon, and the blue portion is a Schwann cell. So the Schwann cell surrounds itself completely around the axon and creates the myelin sheath. Now remember, I said the oligodendrocyte had a lot of these little paddle processes leaving it, and so it could create like 50 different segments of myelin. A Schwann cell itself surrounds an axon in the peripheral nervous system. So one Schwann cell creates one section of myelin. And Schwann cells also can help digest dying axons as well. So if you have an axon that is broken or is just not functioning anymore, the Schwann cell can help digest it and kind of get rid of it. Now, I had already drawn the comparison here between the oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. So I would like to focus on these two a bit more. Basically, in the peripheral nervous system, if we have an axon of a neuron that's damaged and it's kind of like a stump, if it touches a bit of a Schwann cell, it can sprout and begin to grow again quickly. And if the ends of axons are cut, but they're close enough that they can touch in the peripheral nervous system, the Schwann cells can help them reconnect. So basically what this tells us is that if there is damage to the peripheral nervous system, it can possibly be repaired. The Schwann cells can help repair these connections and repair these axons. However, if we focus on the CNS, it is a different story. These axons exist again in the CNS. So The ones that we're focusing on are in the brain or in the spinal cord. And if there is damage to axons in these areas, even if there are new sprouts, even if new sprouts actually form off of axons, if they encounter scar tissue, they can't reestablish an original connection. So if there's damage that happens, it can't be fixed, essentially, or it can't be made the way that it was. So if an axon gets cut, it can't be reconnected again in the central nervous system. Specifically, what happens here in the CNS is that the astrocytes will produce a chemical signal, which those astrocytes are another type of supporting cell that we talked about earlier. They produce a chemical signal for axons in the central nervous system to stop lengthening or stop growing and to start sprouting terminal buttons. But if those axons aren't long enough to make connections or are broken off, this really doesn't serve any functional purpose. So the point here is that damage in the PNS can be repaired. Damage in the CNS cannot be put back together the way that it was. So damage in the brain or spinal cord area is much more serious. And again, this is due to the supporting cells, the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. And I wanted to make a note here about multiple sclerosis. Multiple sclerosis is a disease or a disorder where the myelin surrounding an axon is affected. Specifically, myelin just quits being produced, basically. So you have axons with no myelin. And this is happening in the central nervous system. So this is affecting the myelin produced by the oligodendrocytes. So I think the takeaway point is the damage that occurs through MS is basically not repairable damage because it is happening in the central nervous system and because it's the oligodendrocytes that are being affected. Now let's move on a bit to what is called the blood-brain barrier. You're probably familiar with the blood-brain barrier, at least maybe just the term. What this is a selectively, and that is the key word here, selectively permeable barrier between the fluid surrounding the brain and the blood. So this is a barrier that is kind of protecting the fluid surrounding the brain from the blood on the outside. This is very important because there are some substances that we don't want coming in or out of the brain freely. Now here we can take a look at capillaries that are everywhere in the body except in the brain. And you notice these little gaps here. These gaps permit substances to kind of free flow in and out. A capillary in the brain looks completely different. You notice here that you don't have those same gaps. So many substances can't get through either in or out. And again, this is a very good thing and what we want because we want a very protective layer around the brain. As we saw in the last slide, within the central nervous system, those capillaries really don't have the gaps that exist everywhere else in the body. So what that means is that a lot of substances can't travel through those capillaries. So some substances will bind with special proteins in order to travel through the capillaries. So for example, glucose fuels the brain. Glucose will bind with a special protein to travel through the capillaries and fuel your brain. Also, other substances will bind with special proteins to travel through those capillaries to get rid of toxic waste. So to move things out of the brain that don't need to be there anymore, these substances will bind with special proteins to be moved out through the capillaries. Now, what is the overall point of the blood-brain barrier? It's important to the balance between substances within neurons and those in the surrounding extracellular fluid. Specifically, if that particular composition is messy, even a little bit, it can really affect the transmission of messages that's happening between neurons. Specifically, it can really disrupt them. And if that happens, it will really disrupt your brain function. So the blood-brain barrier is incredibly important for maintaining that balance. And lastly, as we've noted, there are some areas that are more permeable of the blood-brain barrier. And one I'll draw your attention to is the area postrema, which if we're looking here on the right, that is in this brainstem area here. So this is in the older part of the brainstem, so lower down here, area postrema. What this area does is allow for neurons to detect poison in the system. Now, what it does also is that while it is a bit more permeable and allows these neurons to detect the presence of the poison, it also prevents that poison from circulating to the brain. Interestingly, this area detects poison, but it also controls vomiting as well, which makes sense. Why this area would function in this way and why this area of the brain would be more permeable, because it serves a direct function. If there is poison in your system, then it would be beneficial for you to get rid of it. Vomiting is one way to get rid of it. This particular area detects that poison, keeps it isolated from the rest of the brain, and also helps you induce vomiting. So this is an example of an area of the blood-brain barrier that is actually a bit more permeable, but as you can see, it serves a very important function. So that's all we'll talk about in this lecture. Next week, we will continue on with this chapter and we'll focus more on how neurons communicate with each other. So that will be focused more on action potentials. So I hope you all have a wonderful week and make sure to read through this chapter and yeah, have a wonderful week. I'll talk to you soon. Bye-bye.